Hello friends, today we discuss about polymerase chain reaction. Introduction. Polymerase chain reaction is a common lab technique used to make many copies of a particular regions of DNA. This DNA region can be anything the researcher is interested in. It might be a gene whose function scientists wants to understand or a genetic marker used by forensic scientists to match crime scene DNA with suspects. The goal of PCR is to make enough of the target DNA region that it can be analyzed or used in some other way. PCR is used in many areas of biology and medicine, including molecular biology research and medical diagnostics. Now, requirements of PCR reaction. PCR reaction involves target DNA or genes to be amplified. Two primers. Primer, a short sequence of nucleotides, usually around 20 nucleotides, that provides a start point for DNA synthesis. Two primers are used in each polymerase chain reaction and they are designed so that they flank the target region. Take DNA polymerase enzyme, a polymerase enzyme that makes new strands of DNA using existing strands as template. Tag polymerase enzyme obtained from heat tolerant bacteria known as Thermus aquaticus. DNTPs and magnesium chloride act as the cofactor for the DNA polymerase. It provides bivalent cation. Stages of PCR. The key ingredients of a PCR reaction are tag polymerase enzyme, primers, template DNA and nucleotides. The ingredients are assembled in a tube along with magnesium chloride, act as cofactors needed by the enzyme and are put through repeated cycles of heating and cooling that allow DNA to be synthesized. The basic steps are as first, denaturation. Heating of the reaction at 96 degrees centigrade so strongly to separate or denature the DNA strands. This provides single-stranded template for the next step, that is annealing. Cooling of the reaction process at 55 to 65 degrees centigrade temperature so the primers can bind to their complementary sequences on the single-stranded template DNA. And third one is synthesis or extension. Rising the reaction temperature up to 72 degrees centigrade temperature so tag polymerase enzyme extends the primers and synthesizing new strands of DNA. This is the reaction cycle. Now, applications of PCR. Using polymerase chain reaction, a DNA sequence can be amplified millions of times, producing enough DNA copies to be analyzed using other techniques. For instance, the DNA may be visualized by gel electrophoresis, sent for sequencing, or digested with restriction enzymes and cloned into a plasmid. Polymerase chain reaction is used in many research labs and it also has practical applications in forensics, genetic testing and diagnostics. For instance, PCR is used to amplify genes associated with genetic disorders from the DNA of patients. PCR can also be used to test for a bacterium or DNA virus in a patient's body. If the pathogen is present, it may be possible to amplify regions of its DNA from a blood or tissue sample. Limitations: PCR can synthesize only 5 kb of DNA and genes of already known sequences isolated from DNA. Thanks.